Hello everyone. So today it is week 10 of NPTEL TF live session. So today we will be studying about influenza viruses. Sir is already teaching that in this week 10. So I'll just go through a bit about influenza viruses, and then I will go to one doubt which someone has posted last week regarding one experiment. So someone else has joined, so I will just begin with the class. If anyone has any doubt, they can ask me, or they can post in the portal also. So first, before going on to about influenza viruses, we will understand, like, we all know that there are three types of hosts. Host means a person who is getting infected. So infection can target any of these three types of hosts. So first is one resistant host will be there, second is one tolerant host will be there, third one is one susceptible host will be there. So first, uh, like, resistant hosts means we can understand that the host is resistant to the infection. So there will be elimination of the pathogen, or there will be reduction of the negative impact of the infection. Uh, and the resistant host will regain fitness by recognizing and eliminating the pathogen. So it will easily recognize its immune system and it will eliminate the pathogen easily. And the tolerant host will be like, it will be tolerant to that infection, means it won't remove the pathogen from the body, but it will reduce the immunopathology and tissue damage that is inflicted by the pathogens. For example, reduction in tissue damage. For example, in case of the resistant host, it will completely eliminate the pathogen. This, you can see, only one virus is present and it won't be susceptible. It will have a, like, it will also be tolerant to the pathogen. Because it is completely removing, so no other harming of the host by the pathogen. Then it will be tolerant host, where there is reduction in the tissue damage. For example, the in this type of host, they will not completely remove the pathogen, but some amount of pathogen will be there in the body, but there will be reduction in the, and another whole type of host is the susceptible host. That is, they cannot reduce or they cannot eliminate the burden of the pathogen, they cannot eliminate the pathogen, they cannot reduce the tissue damage, but they are unable to tolerate the negative consequence of the immune response to the infection. And what it will lead to the susceptibility of that host towards the infection. So there will be further immune mediated damage or pathogen mediated damage which will make it susceptible to that particular infection. So you also can decide which one are you, whether you are, sometimes some people are like too much attracted towards or too much uh, what is susceptible towards some infection. Some people will be, some people won't have any infection, some people will somehow have infection. So there are different kinds of immune response, different kinds of person who are having varying amounts of immune responses and varying amounts of susceptibility to infection. And so it can be broadly divided into these three types: resistant, tolerant, and susceptible. So then let us jump to influenza. So influenza, which is mostly referred to as flu, we all know flu, whatever we tell like seasonal flu has come, it is mostly caused by influenza or other viruses also are there, rhinoviruses are there and many other are there. But mostly flu means it comes from influenza flu. So it is a contagious viral infection caused primarily by the influenza virus A or influenza virus V. So it affects mainly the upper respiratory organs. For example, the nose, the throat, and infrequently the lungs. But other organs like heart, brain, muscles can be involved. It occurs worldwide and causes considerably morbidity and mortality with pandemic, epidemic, or seasonal patterns. So every season we hear the term flu. Like doctor prescribe that flu has, that patient is suffering from flu or something. So it is all caused by this influenza. So epidemics of flu happen annually during autumn and winter in temperate areas and produce significant mortality and morbidity each year. So uh, you can understand if temperate areas mostly it occurs during autumn winter. In our like tropical areas also it occurs during the not in the winter obviously, and throughout the year you can see this flu thing is going on. So you nowadays you cannot like differentiate like whether it is like weather specific or not. Yes, particularly in winter it increases due to the cold maybe. So how this virus is transmitted? So it gets transmitted from one person to another with respiratory droplets which are produced when the patient coughs or sneezes. And when there is a close contact that is less than 1 meter, it is uh, frequently needed to get infected. So individuals usually recover after a few days, but influenza can give rise to complications and can even lead to death, especially in high groups like pregnant women and those with underlying immunodeficiency state. So people who are like in high risk group who are having some underlying disease or some whether if they are immunodeficient or whether pregnant women, in there this infection can cause certain complications, and the symptoms include high fever, body ache, headache, severe malaise means it will be like it will be pain in the body, then dry cough, sore throat, and runny nose. And it can be differentiated from the common cold, which is caused by the rhinovirus, by clinical presentation. So various ways you can differentiate with the normal flu, like the normal common cold, which occurs every season. So first we will go to the history. Like influenza virus has been known to cause recurrent epidemics of acute febrile syndrome every 1 to 4 years for at least the recent centuries. So it has caused not only epidemics, but first it was reported An influenza-like illness was noted in 1173 to 74. You can understand, so many years ago. Now it is 2024. But the first definitive epidemic was reported in 1694. Obviously, that time people were not knowledgeable about the different kinds of diseases. So, like, some report was there during that time regarding some influenza-like illness. And the first report which actually came into the picture was in 1694. And the greatest pandemic which occurred, obviously, people know what the greatest pandemic which has occurred between 1918 and 1990, where it approximately 21 million deaths were recorded worldwide for this influenza. It was one of the deadliest events at that time in the human history. You can understand 21 million deaths. Like COVID, we all have undergone through a pandemic which is caused by COVID 19, and we know how things went bad when there is so much advancement in the medical field. Still, things went so much bad just 3 years ago. Then you could have understand, like in 1980, 1919, when medical science has not yet that much flourished. No, maybe not that much hospitals were there, not much medicines were there, not much workers will be there. That time, how it has affected the whole world. So, see, after this 1918 and 1919, three other pandemics occurred in the 20th century. For example, in 1957, H2N2 pandemic, 1968 H3N2 pandemic, and 2009 influenza A virus pandemic. So, uh, these three other again pandemics occurred due to this influenza. So you can understand how much scary this virus is. Like it is causing a pandemic, not even epidemic, not even like endemic, but pandemics it is causing. And you all know what are the consequences of a pandemic and how it would have destroyed like millions of people's lives. So in the most recent event, an influenza strains with a combination of gene segments not previously reported in the swine or human influenza virus strains was identified first in Mexico and then in the United States of America. After the involvement of many countries, the pandemic was declared to be over in August 2010. So, in recently, some not recently, maybe 10 15 years ago, another influenza strain kind of pandemic was declared, but not in the whole world, but some, especially in Mexico and United States, which had the pandemic potential, obviously, but it was declared to be over in 2010. So here you can see the history of this influenza virus, and uh, Hi Samyara. So if you had having any doubt, you can raise your hand or even post in the message regarding any assignment or any doubt regarding any lectures.
No, ma'am, no doubt till now.
Oh, okay, okay. So here you can see the timeline of this influenza virus, and before, okay. So this influenza virus can be transmitted through uh, many animals. It is not like only humans can transmit this virus from one humans to another. It is transmitted through marine mammals. Like here you can see some whale or any seal. It can be transmitted from bats, from wild birds, ducks, poultry, pigs, horses, dogs, cats. So you can understand it's kind of a zoonotic disease. It is like getting transferred from animals to humans. So it is not only like a person who is infected will only transmit. It can also get to a person via some animals also if it is a infected. So here you can see the timeline of how the influenza virus has caused the havoc over the years. Starting from 1918, which has the Spanish flu. Uh, so this Spanish flu, which actually almost killed 21 million or 50, almost too much people worldwide. It occurred in 1918 and mostly in USA, Asia, and Europe, and it was caused by a strain H1N1. And here you can see 1930 again. Some H1N1 strain caused this in USA. It caused one epidemic. Then in Europe by human H1N1. So this Spanish flu was caused by the avian virus was the cause for creating this Spanish flu. So from the birds it went to the human population. Then in 1957 again it was caused by another strain H2N2 HA NAPB1 avian H1N1. So these are different strains. So there are different kinds of proteins in the influenza virus. So there are HA, PB1, PB2, then M1, M2. These are the different kinds of proteins which are present in the influenza virus. So all are having different function. We generally name the influenza virus based on this H protein and N protein, and according to the type of the protein they are having, we give the names of the viruses. For example, H1N1 means the particular influenza has the H1N1 subunit protein, and so it is termed as H1N1. So there can be multiple permutation combination of this H1, like the H protein and the N protein, and it can give rise to different kinds of new viruses. So this HA protein is mainly responsible for entry of the virus into the host cell. So this HA or hyaluronic acid present on the influenza virus will uh, go and bind to the sialic acid receptor found in our glycoprotein or glycolipid receptors. In our cell surface there will be receptors present, and there will be glycoprotein and glycolipids will be present in our plasma membrane or the cell surface, and there some residues will be there which are known as sialic acid residues. So with that sialic acid residue it can enter into the our host cell. uh, so this HA is thus helpful in entry of the influenza virus into our host cell. And this Na, what is that? This is neuraminidinic acid. While leaving the leaving this leaving the host cell, like whenever a virus enters, it gets replicated inside the host cell. Then it forms more and more virus particles. Then it leaves that particular host cell and infects a nearby cell. So when it is leaving the host cell, what is happening? This neuraminidase, one enzyme it produces, which cleaves the it also leaves the host cell via the attachment with the sialic acid receptor. So when it has to exit, that neuraminidase will cleave this receptor, this sialic acid residues, so that the virus can completely leave out of the host cell. So both of these are important for entry and exit out of the host cell. So whenever there is a combination of these different HI and NF proteins, it can lead to different permutation combination of different kinds of influenza viruses. For example, here you can see H1N1 was causing some diseases in the initial period. Then it got recombined and formed H2N2 virus. This H2N2 again caused disease in 1957. Then H3N2 it caused disease in 1968. Here you can see it is a different subtype of HF, and it was also caused by avian influenza virus. Then there is in 1970, after that in 1970, it is in Taiwan. Then H3N2 caused some epidemic. Then again H1N1 in Europe in 1976. Then 1977 it was H3N2 again. Then 1977 here also you can see H1N1, another strain came PDM09. Then 1979 H1N1 in Europe. 1980 in Japan, H1N1 as well as H3N2. Then 1980 it is again in Japan, another one. Then 1987 France. So you can see in different parts of the world these kinds of viruses are there. It is not like only at a particular country it is originating, but since it is communicable, like one person to another, it can easily spread like COVID-19. It can easily spread to different kinds of people. If there is a person who is traveling from abroad to a particular place and carrying that virus, it can get transmitted to the people of that region. So here 1987 it is in France, 1989 it is in China by H1N1 H2. Then in 1994 H1N1 in UK as well as swine H3 in UK. In 1998 also it was in USA. Then 1999 it was in USA again. 2000 also, 2000 to 2023 one strain was there, H1N2. Then again at different time periods like 2005 H1N2 is there, 2006 H1N2 is there in China. There's the swine flu. It is a new strain H1N1 PDM, which occurred in India in 2009, then 2011 USA again, then 2012 Australia, this chain, and again USA. So you can see the different timeline of these different kinds of influenza strains which came up with, like COVID-19. We saw like every year it was a new strain coming up. Here in this case also, it in case of influenza also it happened the same. Like new strains are coming up every year. So you can understand, like for a disease like this where so many new strains are coming up, it is very difficult to actually develop any kind of antiviral therapy, because if you are one year you are designing one antiviral therapy against one uh, particular strain, again next year a new strain is coming up, means that viral antiviral therapy will be ineffective against that particular strain. So it is of no use giving like every year, or otherwise every year you have to develop some uh, specific antiviral or specific vaccine against a particular strain. But that is like we all know that how much cost this vaccine or these therapies or these antivirals are needed to produce this kind of things. So it is not possible every year to generate the amount of the vaccines for the whole world, especially for a particular strain. So you can understand how brave or how that these viruses are, like how clever they are. Like they every time change their genetic material, and they every year so that they can escape the our immune system, or like they can escape the um, about like the current medical therapies. Hello, Janvi. Uh,
Yes, ma'am.
you had a query like you posted, I think, last week regarding that Cabot experiment.
Yes, ma'am. I had.
So today I will be discussing about the experiment. So you just tell me what you couldn't understand in that experiment, so I can uh, like tell you about that experiment. Huh.
Actually, it was the basic conclusion of the experiment. It was said that they performed certain things, but I couldn't exactly understand what was the conclusion regarding it. Maybe I'm not aware of the gas or protein estimation that they did about the experiment or
Okay,
something related to it.
okay. So you want me to explain that now? Then I can explain that also. I have actually included that in my slide.
Okay, no problem, ma'am. I'll be joining. I'll be remaining in the session throughout. So whenever if it is included, then please.
Okay, then after this, like after like finish teaching, then I will then explain that experiment.
Okay, ma'am. Thank you.
So first we will go to the etiology of the influenza virus. Uh, Influenza virus belongs to a family of viruses named Orthomyxoviridae. So there are many families of viruses, like different viruses are belonging to different uh, families. I have in the last class, I think I have taught about the different kinds of families and the how they are like named. So influenza belongs to the Orthomyxoviridae family, and it is an RNA virus, means its genetic composition is of RNA material. It does not contain any DNA material. It is a single-stranded RNA virus, and it is having diverse antigenic characteristics. And mainly, they are divided into three types. One is A, one is B, one is C. Mainly, there are three types. Another one is there D, but that is not that much prevalent, and mostly of the epidemics and outbreaks of flu are caused by type A and B, and C is generally responsible for some mild upper respiratory symptoms. So C is also very not that much like relevant in causing pandemics or something. Influenza A and B are the main uh, like outbreaks of flu and epidemics are caused by these two uh, types only. So the influenza virus have spherical or filamentous shape with an envelope containing glycoprotein and a single-stranded RNA genome, RNA gene. And the two most important glycoprotein, as I already told, in the flu virus are hemagglutinin H or HA and neuraminidase or N or Na, and they have important role in the pathogenesis of the disease. So as I told that hemagglutinin is required for attachment of the virus to the host surface. It binds to the sialic acid uh, residues present on the glycoproteins on our cell surface, and neuraminidase is responsible for cleaving the virus out of the cell. So these are important for the entry and the exit of the viruses. And uh, these two can form combination with each other, like as I told H1N1, H2N2, H3N2. These will form combination, like one HA will combine with one Na and form a different kind of strain. So here you can see for influenza type A, there will be at least 16 variable hemagglutinins and 9 distinct NAs. So 16 types of H proteins can be there and 9 different types of Na can be there. N can be there, and with the help of these different antigens, it can be further divided into subtypes on the variable combination patterns of their own specific H or N proteins. For example, as I told H1N1 or H3N2. And in the nomenclature of these viruses, other variables such as the place of initial isolation of the year of isolation are also included. For example, there are many viruses which are suppose uh, according to the name, the virus name is given according to the like this H and N subtypes, and then again on the place of origin. If it is human origin, it will be given human. If it is swine, means pig, so coming from the pigs, it will be swine origin. Then if it is from the birds, it will be of avian origin. And also when it was first isolated, for example, there are strains like New Mexico strain. There will be strains from Puerto Rico, PR8 strain we call. So depending on the place of isolation, these viruses will be named. And influenza B also has a similar structure to type A, but due to fixed antigenic characters of HA and NA, there are no subtypes in the virus, because in this influenza B virus only one subtype is there, and there are not like HA and NA subtypes. So there cannot be divided. So this influenza B also is kind of mild only compared to A. So still it was in 1970 some small antigenic variability was seen with the virus, uh, and it was seen that they have divided into two antigenically distinguishable lineages. So here you can see this is a table with the different types of influenza, like influenza A, influenza B, C, and D. Mainly influenza A is the one causing these pandemics, and it can infect a wide range of animals. Obviously, you can see how many H and Na proteins are there. So you can understand like how many subtypes it can form if you do a calculation of the permutation combination and uh, this. And I think this I took from a book, and it is written 16H to 9 distinct Na, but I think now it has increased also because the book was not up to date. Maybe it was maybe 2 3 years ago. It is now maybe increased to my knowledge 11 Nas and maybe 18 Hs. So you can understand how much uh, like it is uh, it can like create a number of strains. So influenza B mainly infects humans. Influenza C also infects humans and pigs, but more rare than type C and B. It will only cause some mild disease. And influenza D is not at all human. It infects only cattle. So there are subtypes in case of A, like avian influenza can also infect humans. So that Spanish flu was originated from avians or birds only. So here you can see H5N1, H7N3, H7N7, H7N9, H9N2, H10N8. These many subtypes can be there. Then in case of swine, it can also infect humans. For example, H1N1, H1N2, H3N2, and most common human subtypes are H1N1 and H3N2. And HPAI versus LPAI means highly pathogenic avian influenza versus low pathogenic avian influenza. Means they can be divided as high pathogenic or low pathogenic based on the specific ability of the virus strain to kill the chickens in the lab setting. So these are, if you like, if they are highly pathogenic means these viruses can kill the chickens in the lab setting. In the lab setting, they will infect the chickens or the small baby birds, and they will see whether it is capable of killing that uh, killing that or not. I know it is not sounding good as I'm telling, but this is how experiments are done to actually prevent things in future. And like HPAI H5N1 means it is highly pathogenic. That means when you are treating this particular thing in the chickens, it will kill the birds. And there is also strain of low pathogenic avian influenza H5N1. So there are both high pathogenic avian influenza as well as low pathogenic avian influenza. So when it is not able to kill the virus, then it is a low pathogenic one. So epidemiology means uh, how it is affecting the, like, how it is affecting and the population, kind of. So it occurs in district outbreaks of varying extension and intensity every year. And this epidemiologic pattern is based on multiple factors such as changing nature of the antigenic properties of the virus, then how it is getting transmissible, like the transmissibility power of the virus and the susceptibility of the population. And the susceptibility of a particular community is one of the most important factor in strength of the epidemics and its mortality or morbidity effects in specific. For example, it was seen that in Iran, due to there was partial immunity to a particular group of people. So, when a pandemic occurred in Iran, the country did not have a high mortality rate. Maybe people were infected, but the but most of the people did not have a high mortality rate. So you can understand that it depends on the population also, and it depends on the genetic factors also of how a people will get affected or not. Uh, so so pandemic obviously means like whenever there is a whole country or a whole world is affected by a particular disease, then only it is called a pandemic. But pandemic doesn't mean that there will be a high mortality rate. So in here it was seen that in Iran, the country did not have a high mortality rate even though there was a pandemic. So it depends on the particular population also. So the influenza virus has 8 segments on its uh, its genome is divided into 8 segments. It is not like a whole stretch of genome which is transcribed and translated, but it is a whole. It's all its proteins are segmented into eight parts. So all the proteins are encoded by a particular part. So it is having a segmented pattern. So since it is having a segmented pattern, you can understand there is high rate of reassortment on its genome. And the emergence of pandemic strains usually has been caused by animal and human type reassortment and the antigenic shift. Uh, so uh, So there is a term which is known as the antigenic drift and antigenic shift, which I will tell later. So antigenic, so there, uh, it is between the years of the antigenic shift. Antigenic drift happen annually and have resulted in outbreaks of variable extent and severity. However, antigenic drifts are usually less extensive and severe than epidemic or pandemic associated with antigenic shifts. So antigenic drifts are resulted from point mutation in the RNA gene segments that are responsible for hemagglutinin or neuraminidase. Accordingly, they occur sequentially as the virus spreads through susceptible populations. And influenza usually has the highest attack rate among young people, while high mortality rates are reported among older adults. Obviously, in older adults, the immune system is much suppressed compared to the younger people. So, even if the younger adults are more susceptible, as I already told, they are kind of this tolerant type of to a particular infection. But in case of the older people, since the immune system has also become weakened, they become a susceptible host compared to the young adult. So, in addition to the elderly, mortality and morbidity are high, in especially those who are having a high risk medical condition. For example, extreme of ages, like too too much aged they are, or they are having any cardiovascular disease or any metabolic disease such as diabetes. And obviously, as I told, pregnant women or immunocompromised people are more susceptible to this kind of diseases. And during pregnancy, also, it was observed in 2009 pandemic. It was observed that the people who were pregnant, the increased rate of influenza morbidity and mortality were high in those specific people, because in pregnant people the immune system will not only is not uh, that much strong, or it will be like the immune system will also protect the fetus, so it is kind of shared between two people. So That time also it was seen in 2009 that these people who were pregnant that time were also very much in high risk. And also from data from different pandemics also suggest that in the influenza complications were higher in the second and third trimester of pregnancy compared to the first trimester. So here are the groups of at risk who were in case of influenza infection. So first it was unvaccinated infant. Obviously, once a person is born, they has to undergo a list of vaccine. Like they had to take a list of vaccines against different kinds of diseases. So in case of unvaccinated infants aged 12 to 24 months, uh, they were very much susceptible. Then persons who were having asthma or any kind of chronic pulmonary disease, such as cystic fibrosis in children or chronic obstructive pulmonary disease in adults, they are also very much susceptible. Then person who are who has this cardiac disease, cardiovascular disease, they are also susceptible. And then people who are having immunosuppressive disorder or who are receiving some immunosuppressive therapy, for example, the HIV infected people whose immune system uh, is destroyed kind of by the HIV, they are also very susceptible. Then people with sickle cell anemia and other hemoglobinopathies, where there is a problem with the formation of the red blood cell or hemoglobin, they are also susceptible. Then people with disease which require long-term aspirin therapy such as rheumatoid arthritis, they are also susceptible. Then people with chronic renal dysfunction, then people with cancer, then people with chronic metabolic disease such as diabetes mellitus, or with neuromuscular disorders such as seizures, cognitive dysfunction, and also which may compromise the handling of respiratory secretion. And there are adult aged who are more aged, like above 65 years, whose immune system has weakened. Then people who are uh, residents of any kind of age of nursing homes or any other long-term care institution. So these are the more vulnerable people who has a higher risk of developing this influenza like complications. But in people who are like not like us, like adults who are not very much immunocompromised or they are not suffering from any of the diseases, they are more or less normal, or they are not much affected by the influenza. But as we all see during the COVID also, it was a similar case, like people who are having different kinds of diseases, already existing illnesses. They were more susceptible to the COVID compared to the normal people who were not having any disease. They were like viruses were uh, attacking those people who were not diseased, but they were getting recovered soon compared to the people who are having any kind of illnesses. So, as I already told, this is influenza is a flu kind of thing. So, it affects the upper respiratory tract, and it occurs in the respiratory secretion of the patient suffering from influenza, and large amount of viral load are present there, and they can easily transmit this infection to the other person. I told, like, whenever there is a distance of less than 1 meter between two person, they can easily transmit this infection to the other individuals by sneezing or coughing. And it is seen that they transmit this disease via large particle droplets that is greater than 5 mu. And as you know, the more the large particle of droplet is there, it won't remain suspended in air for a long time. It will go and get settled at a particular place. From there only it will spread. And because smaller particles can easily travel through the air. So airborne transmission is therefore not often considered for disease spread, because mostly it is like greater than 5 mu, means these are larger particles, and these will settle down at a particular place, and by somehow, if a person is going and touching that thing, and he or she will get infected, and he or she can transmit the infection to the other person. So, small particle respiratory droplets, which become aerosolized, can stay suspended in there for a long time. Also, can contain the influenza, but it is less compared to the larger particles and can potentially cause disease spread. In a recent study, aerosol transmission accounted for around half of all the transmission events. This suggests the activities to reduce transmission by contact. A large droplets may not be enough to control the transmission of the influenza A virus in household or communities. Uh, so you can understand, like, it is not very much, we cannot control the spread of these droplets from one place to another. So it is not that much controllable. And also, contact with some contaminated surface containing respiratory droplets is another potential source of disease transmission. In adults without other underlying disease, the shedding of virus starts from 24 to 40 hours, 48 hours before the disease is occurring, and the shedding stops after 6 or 7 days, according to most studies, and after 10 days, according to most of the investigation. It should be considered that longer periods of shedding and infectiousness can occur in children, elderly adults, immunocompromised hosts, and patients with uh, chronic illnesses. Uh, so you can understand, like, it takes only 24 to 48 hours for the virus to shed from a person. Like, shedding of virus means that when a person is releasing the viruses from their body. For example, in case of COVID-19, it was seen that they used to, people with this infection used to shed the virus for about 10 days. And in case of this influenza, you can see that it starts from 24 to 48 hours after they are being infected, and then the shedding stops after 6 to 7 days. So, according to the age also, or the severity also, they can start shedding. Means more and more of virus will come out of your body, and it can infect the other person. So, and it also can, you can also get this disease via contaminated surface, as during COVID-19 also, you like, from the surfaces, like, there was so much, um, like, people were cleaning the surfaces with those, uh, what to say, the alcohols, kind of, whatever the uh, things were there, and then with the detergents and everything. So, since it is a aerosol, through aerosol transmission, you can get these viruses. So, this contaminated surface is one of the most potential source of disease transmission. So here I have already shown, like, how influenza gets transmitted not only through humans or avian, but also through pigs, horses, dogs, cats. Every animal can actually, all these animals can actually get infected the influenza virus, and it can get transmitted to the humans. So any kind of doubt is there?
No, ma'am.
Okay. So there are many symptoms which overlaps with other diseases. For example, high fever, muscle ache or joint pain. There will be chills, runny or stuffy nose, dry cough, fatigue, tiredness, diarrhea, vomiting, nausea, sore throat, headache. These all symptoms, you can see, it is common to many types of diseases. Even in COVID also it was similar. Even in common cold also you can have similar symptom. But how will you know whether this is influenza or common cold or something? So if you are a perfectly like a person who is having no disease kind of, then it is fine, no problem. But sometimes people with underlying disease often mistake this like they are having some common cold and do not visit the doctors, and they undergo like so much like what to say, the symptoms worsen and they ultimately there are consequences. So whenever some person is having any underlying disease and they are having like high fever for this so many days or this kind of symptoms, then they should actually visit doctor and get checked whether it is common cold, whether it is what kind of virus is there, because they will give some test to know that whether it is influenza, whether it is COVID-19, whether it is any common cold or not. So it is advisable to everyone to go to the doctor whenever there is the symptoms are not getting healed in 2 days or 3 days. So influenza, as I already told, it has been responsible for some of the worst pandemic in history, and it was the largest pandemic worldwide epidemic in recent history, like 1918 to 1919, that Spanish flu, and it has death has reached 50 million in less than 1 year. You can understand how many people died in this one pandemic, and compared with the roughly 10,000 to 15,000 who die yearly, like every year there is a death. You can understand there is a death every year by influenza, and we do not even know about it that this much 10,000 to 15,000 death is also a good number. So every year there is a death of about 10,000 to 15,000 of people every year by this influenza, and in pandemic it almost reached 50 million in less than 1 year. So, as I already told, there are different kinds of antigens like Na and Ha, and in the and variation in the structures is generated by two different mechanisms: antigenic drift and antigenic shift. For example, antigenic drift involves a series of spontaneous point mutation that occur gradually, resulting in minor changes in H and Na over time. Means point mutation means it will only affect one kind of nucleotide. So whenever there is a point mutation, there will be minor change. It is not like a too much change at one time. So uh, it is a minor change that occurs over the time period. Spontaneous minor changes, point mutation, that occur gradually. But antigenic shift is the like is it is sudden emergence of a new subtype of influenza where the structures of H and Na are different from the virus present in a preceding year. So from the last year to this year, there is a sudden change or a sudden new subtype of influenza that emerges, and there is a complete change in the structure. So this antigenic shift is more like fast and rapid compared to the antigenic drift. So here you can see there are two mechanisms that generate variation in influenza surface antigen. Here you can see it is a host cell. This is person A, year 1, and you can see this antigen, this Na and Ha is there. So this is infecting this host cell, person A, year 1, and this is uh, infecting that host cell. But over the year, what is happening? Uh, it is changing its structure. Like it will take years to do the antigenic drift because there is point mutation. Here the genome also you can see is different. But over the years it is changing one by one. It's new, the like the what to say, the nucleotides in its genome. So it is constantly producing different types of H and Na. So at last, after some years, like these viruses also will be viable. These will also infect the other people. So here you can see another, in the subsequent years, it will create a new virus. So here you can see this virus was not being able to infect the host cell. It cannot go and bind because the antibody is going, the antibody going and binding and neutralizing the virus. But the virus, as it is changing its structure and its genome, then this virus can easily interact with the salicylic acid here on the cell surface, and here it can infect the person A over subsequent years. So, in antigenic drift, the accumulation of point mutation yields a variant protein that is no longer recognized by our antibody to the original antigen. So, if in the previous years you are not infected by the same virus, that particular virus can change itself and can infect you in the subsequent years. But this is a slow process and it can take years. But in antigenic shift, what happens? It occurs very rapidly. So here you can see a person. It is a non-human host cell, but it is, suppose, a pig. So here it is infecting a swine. Influenza is infecting the pig, and a human influenza is also infecting. But it will not be susceptible to the human influenza. But what happens? Infection is occurring at the same time. So two viruses will come and enter into the host cell, and they will release their genome into the host cell. So from there, what happens? It will these two genomes, they will reassert among themselves. They will form a combination. And here, what happens when they are coming out of the cell? They are creating a new virus where you can see it is a combination of both the human influenza as well as swine influenza. So you can understand like how it is dangerous, like how two different host pathogens can actually combine together to form a new kind of strain which might be more threatful to both the host, or it might not be. So you can see like antigenic shift may occur by reassortment of an entire single-stranded RNA between human and animal variants infecting the same cell. So this single-stranded RNA will combine together and it will create a new kind of virus. And the surface proteins of the new influenza subtype are so different that human antibodies no longer can recognize them, and humans thus have no immunity. Only two of the eight RNA strands are depicted. Since it is like eight stranded, since it will be eight strands will be there, and here they have only shown two strands like HA and NA, suppose. So this HA and NA, this human influenza is having a different HA, this swine influenza is having a different HA. So what if this HA of human influenza and NA of swine influenza is mixing together? So it will form a mutated kind of virus which again can affect the human cell. And our antibody cannot recognize obviously that particular uh, antigen on the surface of the influenza because we don't have that antibody yet. So you can understand like this antigenic shift and antigenic drift will be uh, causing too many new strains of viruses which will actually uh, which can actually cause these kinds of pandemic and epidemics as well as endemics. For example, in COVID also you could have seen many viruses, many new strains are coming up every day because there was also, and one of the things which these RNA viruses have is they lack the proofreading activity of the polymerase. For example, they require their genome to replicate. So they need a polymerase which will increase the number of their genome. So there will be these viruses do not have proofreading activity. So randomly they can put any kind of mutation. Like our genome has proofreading activity, our polymerase has proofreading activity. So it will be like they can know whenever there is an error, they know they will rectify it. But in these kinds of pathogens, there will be no proofreading activity, and they will continue to add wrong nucleotides, and they will replicate inside the host cell with that wrong nucleotides, and they will create a new virus. Yes, yes. Okay, 1 minute, I will check. Can you see it now?
Yes, ma'am. No, there is no problem actually. Your voice is clear and everything is seen clearly. But the problem is that whenever you're updating or shifting to the new slide now, it's already stuck on the previous one. So there is no meaning of just listening to and not being able to see the content. That's why.
Okay, so you now it is visible. I am on
Yes,
slide
ma'am, you are
14.
two mechanisms generate variations.
Ha ha, yes, yes.
Yes, sir. Yes, ma'am. Now it's visible.
Okay, okay. I think there is some network issue. So there are these two mechanisms by which this influenza virus create different kinds of subtypes, different kinds of strains, which actually cause this pandemic. So from the as in the case of Spanish flu, we saw that that virus was originated from the birds, but then it infected the whole human population, which actually removed 50 million people in less than 1 year. So uh, the immune response thus contributes to the emergence of this antigenically distinct influenza strain. Obviously, as I already have told you before, like our immune system wants to remove the virus from our system, but the virus wants to stay in our system. So the virus is also creating newer and newer number of different kinds of strains so that it can bypass our immune system. So our immune response is also one of the factor which is contributing to the emergence of this antigenically distinct influenza strains. And in a typical year, the predominant virus strain undergoes antigenic drift, generating minor antigenic variants. And as individuals infected with influenza mount an effective immune response, they will eliminate that strain. So, whenever there is an effective immune response generated against the influenza, it will be eliminated from the system. However, the accumulation of points mutation sufficiently alters the antigenicity of some variants, so they are able to escape our immune elimination and become a new variant that is transmitted to others, causing another local epidemic cycle. So, from suppose in my system some influenza virus is entering and it is causing a new kind of strain and coming out of it is coming out of my cells and through my coughs or sneezes, it is going and infecting another person nearby to me. So that person is infected with that particular new kind of strain. So that new strain can cause new kinds of symptoms, new kinds of consequences can happen, which occurred compared to the previous strain. So, the role of antibody in such immunologic selection can be demonstrated in the laboratory by mixing of influenza strain with a monoclonal antibody specific for them for that strain and then culturing the virus in the cells. The antibody can neutralize all unaltered viral particles and only those viral particles with mutation can result in altered antigenic escape neutralization and able to continue the infection. And within a short time in culture, new influenza strain emerges just as it does in nature. So in the laboratory setup, you can easily identify if some new strain is coming up by influenza. For example, if you can have a influenza particles in your lab, you can easily like synthesize antibody against that by of it requires a mice model. Like you can infect the mice, you can take out the serum from the mice, and then you can get these antibodies specific for that strain, and then you can incubate that influenza uh, strain with a particular antibody, and what happens, that antibody will neutralize that particular influenza strain. So that antibody can neutralize the particular influenza strain. But if suppose there is some mutation and some new strains have come up, then that antibody cannot go and bind to that particular new strain. So what happens? Then antibody can neutralize all the unaltered viral particle, and only the viral particles with mutations will survive because they will escape this neutralization procedure by the antibody and able to continue the infection. So, within a short time in culture, you can identify a new influenza strain like that, how it emerges in nature. So, episodes of antigenic shift are thought to occur through a different mechanism. Primary mechanism, as I already told, there is genetic reassortment between influenza virus from humans and those from various animals. And the fact that this influenza genome contains 8 separate strands of ssRNA makes possible the mixing of these various strands of human and animal. And within a secondary host cell infected with both viruses, there is shuffling of the DNA segments derived from the animal and human cells. For example, in pigs and birds can harbor human influenza viruses as well as their own and sometimes other species. So as I already have shown you that picture, so there can be mixture. For example, a pig, suppose in a bird, it can have its own influenza virus, it can have its human influenza virus, it can have pig influenza virus. So what happens? There will be a mixture of all these influenza viruses, a new kind of chain will come via genetic reassortment, via antigenic shift and drift, and it can cause uh, pandemics. So that is why it is known as swine flu or avian flu, because from the swine or the means the pig or the birds only it comes, and all the proteins that are encoded on one of these mini chromosome derived from the animal are frequently new to humans, which we are having no immunity against. So there will be pandemic or epidemic which can go on. So um, So imprinting of a memory response can influence the susceptibility to future viral infection. Immunological memory, we all know, is an amazing and beautiful thing, and for pathogens that don't change much from one encounter to the another, it can provide us with a lifelong protection. For pathogens which are not undergoing this reassortment or something, like it is stable in its structure, then what happens when it is infecting in our body, there will be the memory B cells which are created in our system, which can remember that this is a pathogen which has already attacked the host cell, and they will give us a lifelong protection by the whenever there is a second time infection. But in case of the pathogens where there is a this kind of mix and matches of this genetic reassortment or this antigenic shift drift or whatever, then there it is not possible for our memory to recognize the pathogen every time. So preformed immunity can come with caveats for pathogens that have evolved mechanisms that allow them to vary their antigenic structure. During secondary encounter with a pathogen that bears a strong molecular resemblance to an agent seen in the past, example that is already they have our adaptive immune system has seen the epitopes and have somehow uh, they remember like yes it is a already I have seen this pathogen. Memory cells specific for previously encountered uh, epitopes are engaged rapidly and efficiently. As long as the cells and their products like antibodies can dispatch the pathogen efficiently, there is no need to mount a primary response to any new epitopes carried by that pathogen. The presence of antibodies attached to a pathogen, either as residuals from a recent infection or produced by deactivation, uh, produced by deactivation of memory B cells will divert naive B cells from responding. So, whenever there is presence of the naive of the memory B cells, they will not allow the naive or the new B cells to respond. They already there is there are antibodies which are present against those pathogens in our body, so they will attack that pathogen. So there is a term which is known as antigenic scene. Antigenic scene is a term used to describe the observation that we rely first on memory responses before activating naive lymphocytes. In other words, when we encounter an infectious agent with some epitopes for which we have memory, we enlist these memory cells are affected rather than activate new cells that target new unique epitopes present on this infectious agent. So you can understand like whenever there is a new pathogen coming and they can see like somewhat I have seen this pathogen but not fully, they will try to activate the memory cells first rather than activating the new B cells to mount an immune response. So that will take some time because suppose your memory B cells are getting activated, then it is deciding no, this is a completely new pathogen, so my new B cells has to get activated. So that will take more time. So for example, here you can see it is a primary response is there. Two types of response. One is primary response and one other is secondary response. So here it is a new B cell. First time pathogen is coming. So new B cells or naive B cells are activated and they are producing antibody specific to these epitopes present on the pathogen. And that same when there is a during a secondary response to the variant of that pathogen, that memory B cells specific to epitopes encountered in the past are getting reactivated and they will help in eradication of the pathogen. Here you can see the already the memory B cells are there which remembers the pathogen and it will activate or it will bind to this epitopes. It will recognize that epitopes they will be reactivated and they will help in eradication of the pathogen. And there are FC regions of the antibodies which will bound to the surface of the pathogen. Here there are Fc regions which will bound to the surface of the pathogen and they will in turn bind to the Fc receptors present on the naive B cells and inhibit them from responding to new epitopes on the pathogen. So here you can see memory B cell activation, antibody production and pathogen eradication. And here uh, even if there is like naive B cell is binding to the new epitopes, then what happens? The Fc region will bind to this uh, new epitopes and then they will bind to the Fc receptors and then there will be a negative signal. So no need to uh, extra add the immune kind of response. So there is a term, as I told, antigenic scene, which is the inhibition of primary responses against unique epitopes on pathogens that elicit memory cell response is called original antigenic scene. So no immune response is mounted to each new epitope during subsequent exposure to the pathogen until the pathogen expresses significant number of unique epitopes and memory cells can no longer eradicate the organism. So in this case, a new primary response is mounted, leading to adaptive effects associated with symptoms and resolution of infection. For example, As I already told, this point mutation occurs by antigenic drift in the influenza, and suppose it is a gradual mutation, so one by one it is changing its nucleotide. So it takes years to develop a completely new strain. So whenever there is a like change, uh, whenever there is a like change in the uh, epitope of the influenza virus completely, so that then only it will activate the uh, then only it will activate the uh, new B cells. And if it is like minor changes which is occurring over the time, then our memory cells can easily recognize, and there will be negative signals whenever there is our naive B cells are going and binding to the surface epitope of the unique antigen. Suppose out of 10 and 8 antigens, one is new and 7 are old. So in that 7, it our memory B cell can recognize. Then that there will be a negative signal which will prevent any new kind of B cell to activate in response to the new antigen. But whenever there is kind of one old antigen and 7 new antigen, then the whole thing becomes a new kind of pathogen. So it will then activate a naive B cell response. So this imprinting of a memory response is important as can influence the susceptibility to future viral infection. For example, here you can see antigen epitope specificity. There are different types of epitopes A, B, C, D, E, F. In first infection, there is a primary response. Here you can see A, B, C are there. Three types of antigens are there. But in case of second infection, there is only C is changed. C has maybe it has changed to another epitope D. So here still there is a memory response because two antigens it can easily recognize from the memory. Like these are the old antigens which are present. So memory response is already there against this A and B antigen. So there will be a memory response in this case. But in a third infection, the B also got changed and it has changed to E. So two antigens are new and one antigen is old. So here what will happen? It can still recognize what in from the last one. It can still recognize the memory. Memory will be activated and it can still recognize that some portion is still I have encountered once. Our cells will recognize it, and then in the fourth infection where all the three has changed F, E, D. So it cannot find any common thing. So it will be like a new kind of response will be generated. So depending on the memory response, it can influence whether it is susceptible, our system is susceptible or uh, uh, non-susceptible to a particular virus infection. So then how our system recognizes what is the response when there is like the attack of an influenza virus in our system. So there can be multiple ways, and here you can see this is a macrophage, and there is this is a cell. This is a whole cell which has been infected with influenza, and here this cell is dying because the viral load is high in that cell and the virus wants to come out. So whenever there is the virus wants to come out, there will be lysing of the cell. So what happens when the virus is dying? So the cell is dying due to the virus. Uh, it can be removed by the macrophage. For example, this is an infected cell which is on the verge of dying, and it is being taken up by phagocytosis by macrophages to for recognition of the double-stranded RNA. Here you can see the double-stranded RNA, which is recognized by the TLR3, which leads to the activation of the NF-kappa B pathway, type 1 interferon production, inflammatory cytokine production, and which leads to the degradation or the removal of the pathogen from the body. So here I hope you all have studied because Sir has also taught these pathways, different pathways, and this viral RNA is released from the virus and there is a degradation. Here you can see in the phagosome this viral membrane and capsid is degraded within the endosome, and the ssRNA is also recognized here by the here is another it is in macrophage where TLR3 is recognizing this double stranded RNA. Here it is in dendritic cell where you can see the influenza can enter via endocytosis, via plasma membrane. There is a fusion of the membrane of the virus as well as the fusion of the membrane of our host cell, and they enter through a process known as endocytosis. And there are formation of these endosomes where there is presence of this TLR7, and there is degradation of the capsids and the membrane of the virus, and this viral RNA is released, which is recognized by TLR7, which then induces the NF-kappa B dependent genes from the NF-kappa B endosome and IRF7 activation from the IRF7 endosome, which leads to the production of this type 1 interferon stimulated genes, these pro-inflammatory cytokines and other things. So this is one case, and this is a complete kind of pathway. Here you can see where the virus is entering via receptor mediated endocytosis. Its hyaluronic acid is sorry, not hyaluronic, the hemagglutinin is getting attached to the sialic acid present on the glycoproteins on the surface of the cell. It is getting internalized in the endosome. Then there is a release of this ribonucleoprotein, viral ribonucleoprotein, which is then entering into the nucleus. It is entering the ER here, and here you can see there is ribosomes attached to the ER, you all know. So there is mRNA synthesis, RNA replication, which occurs here, and no, sorry, this is nucleus only, and this is the ER, and there is a synthesis of the proteins here. Ribosome will be there here, and so the mRNA synthesis and RNA replication occurs inside the nucleus. After which there is this M2 is a protein which is present in influenza, which helps in the ion this acts as an ion channel, and there is a proton flask which occurs, which activates this NLRP3 inflammasome. So here in the cytosol, this viral RNA is recognized by the RIGI. Here you can see the in the cytosol viral RNA is recognized by RIGI, which activates the MAVS or IPS1 pathway, which leads to the activation of cytokines and type 1 interferon. Then here the M2 is a protein which helps in the transport of the proteins and other influx of the proton flux from the nucleus, and then it stimulates the NLRP3 inflammasome formation, which ultimately leads to caspase 1 activation and release of the different kinds of cytokines like IL-1 beta, IL-18. Then in case of another like this PB2 is present, which PB2 will then here you can see these are the PB1 and PB2 are the polymerase component of the influenza virus, and here there are fibrils PB1 F2 fibrils which accumulate in the phagosome and results in the activation of this NLRP3 and the release of this pro IL beta IL-18 cytokines. So these are the innate immune response to influenza virus mainly. So apart from this, uh, there is innate resistance is confirmed, as I already told, by type 1 and type 3 interferon that are stimulated. Here you can see influenza virus is infected, the cell is infected. This alveolar macrophage, as influenza is a upper respiratory virus, it infects the alveolar cells much easily, and this is alveolar macrophage containing the virus which activates the Rigi. Then Rigi will activate this type 1 interferon, and this single stranded RNA is sensed by TLR7. Then this type 1 interferon will act on most cell, and there is secretion of the type 3 interferon also, IFN gamma, IFN del lambda, which acts on the epithelial cells to block viral replication. Here you can see IFN lambda is acting on the epithelial cell. There is also IFN gamma, which is produced by NK cell or natural killer cell. And so the dendritic cell or macrophages that are infected with influenza virus release IL-1 beta. You can see they release IL-1 beta. Uh, Here you can see IL-1 beta is released, which is interacting with the IL-1 receptor present on the dendritic cells, and there is migration of this dendritic cell to the lymph node where they activate the T cell. Then this T cell produce IFN gamma granzyme, which is thus creating adaptive resistance. Then there is also activation of the NLRP3 inflammasome, which also increase disease tolerance by promoting tissue repair. Then T cells, as I told, they secrete IFN gamma to induce antiviral state or induce the granzyme mediated. This granzyme will directly lyse the cells, virus infected cells. And B cell, it will stimulate to secrete antibodies against the virus to mediate adaptive immune response on the host. And there are also inflammatory cytokines such as IL-1, TNF, and IL-17 that are induced as a result of innate signaling, which can also lead to pathology uh, when the duration and extent of cytokine release is increased. And negative regulator of cytokines are an inflammatory cells such as IL-10 and CD200 and CD200 receptor are also expressed to decrease the inflammatory consequences, while positive regulators of tissue repair such as amphiregulin TGF beta are also secreted to uh, like create the homeostasis state. So there are positive regulators, there are negative regulators which are secreted. For example, here you can see the alveolar macrophage, there is which is secreting the receptor CD200 receptor. There is secretion of the negative regulator, the CD200, which is interacting with this, and then there is inhibition of these inflammatory mediators, which is like too much excess inflammation is not good for the host cells. So you need to stop the response also. These are done by these anti-inflammatory cytokines. And there are also, obviously, when there is a invasion of pathogen, there will be tissue damage in that particular place due to the different kinds of mediators. So there will be different kinds of regulators like TGF beta will be secreted to promote the tissue repair which has been damaged. Then there are other like amphiregulin which will also help in the tissue repair. Then this neutrophil will be there, which is this gamma T cell will actually activate this neutrophil, uh, which you all know the neutrophil is the first a cell which goes to the site of infection. So there are positive regulators as well as negative regulators, which actually depends whether a particular host is susceptible to a disease, whether he is tolerant to a disease, or whether he is uh, like uh, has succumbed to the disease. So if there is an excess kind of inflammation by the immune system and there are no positive regulators, then that person can succumb to the disease also, which might be the case for the people who have who actually who actually died in this kind of pandemics. Maybe there is an excessive inflammation and there were no positive regulators to control the excessive inflammation. So it depends on these regulators on what time uh, they are like on and they are off, and whether the host is succumb to in disease or whether it can enter a state of tolerance. So Janvi, so now you can ask me any question because now I will clear your doubt which you have asked, like whether despite watching you couldn't understand about the Tiselius Cabot experiment. So this is the experiment you are talking about, no? Which I am showing.
Yes, ma'am.
Huh. What you could not understand how this was performed.
Yes, the interpretation was that they found different antibodies for different. For example, what is this gamma graph and how is this having certain raises and lower downs? I know that's not so important. We just have to remind that it's serum protein electrophoresis. But still, I had some problem regarding the conclusion of this experiment.
Okay, so what happened was, people knew that there is something present in the serum which is actually uh, acting towards the antigens, which is actually protecting. Like, for example, what they did in this experiment was they took rabbits and they have incubated or have immunized the rabbits with the ovalbumin. Ovalbumin is the egg white which we see that contains the protein albumin. So they have inserted that albumin, which is a very potent antigen. Means some people are very much allergic to this and albumin or eggs kind of, because they are very much allergic to this albumins present in the egg. So they become allergic to eggs. So this is a very kind much potent antigen, and so to see which fraction is actually responsible for creating a immune state in our body, they have like took rabbits, they have immunized with the ovalbumin, then they have collected their serum because they knew that some component of the serum is actually responsible for the like the neutralization of some kind of antigen. So they have taken the serum and they have first divided that serum into two parts. One part, what they have done is that but one part they have electrophorest and have separated the serum proteins according to the electric charge and mass. So here this blue line you can see is the first part which the electrophorest. So they found, like, depending on the charge and the mass, the proteins will get separated. When you are running a gel, it will, depending on the electric charge and mass, it will get separated. So they saw that there are four peaks they got, means four different proteins which were abundant in the serum. They got that protein. Obviously one will be the albumin protein which is present in our body. That is the most abundant protein in the serum. So one protein they got a big peak. So it was obvious that albumin was the most abundant protein in our serum. Apart from that, they got three other peaks. Two were almost similar peaks. So they just gave the name globulin to that. Okay, these apart from this albumin, these other three peaks where they gave globulin name. Then they saw these two peaks are similar, almost similar in size. So they gave one peak as alpha and another is as beta. So these were two proteins, alpha globulin and beta globulin. And another was also present in abundance, which they gave the name as gamma globulin. That is this one. So after that, they only knew that these four proteins were the major proteins which are present in the serum. They did not know up until that which is responsible for what. Then another part of serum which they collected from the rabbits which were immunized with ovalbumin, what they did, they mixed it with that particular serum fraction, they mixed it again with ovalbumin. So what happens when an antigen is reacting with an antibody, then that that fraction will become heavy and they will precipitate. There will be precipitation of the antigen antibody if you are mixing the antigen and the antibody together. So when they mix that other serum portion with ovalbumin, what they saw was one fraction has precipitated. Then they ran that portion, like the other portion which has precipitated, they discarded, and the other portion they ran in the gel again. And then they saw that these three peaks are similar to the previous peaks. Only this blue gamma peak has reduced. So they concluded from the experiment that albumin was already present in the second also. This alpha and beta globulin also are present in the second fraction, but this gamma globulin was missing from that fraction. So they understood that this gamma globulin somehow reacted with this ovalbumin antigen and there is a precipitation. So in that fraction, that gamma globulin was decreased. The amount of gamma globulin was decreased, and from that they understood that this is like a this gamma globulin is responsible for the reacting to the antigen. You understood?
Yes, ma'am. I understood.
uh, this is not by how they just identified that something is there present in this fraction of the serum which they termed as gamma globulin, and they found that this is responsible for interacting with the antigen. After that they wanted to know this, they took this fraction and they wanted to know about the structure, so they digested that fraction, this gamma globulin fraction, with those different kinds of enzymes, that um, papain, then that pepsin. They cleaved and they found out what are the different like how that antibody, particular antibody, looked and what are the different structures. So this
Okay,
gamma
ma'am.
globulin is composed of all the immunoglobulins and the antibodies that bind to the invading pathogens. So from this experiment, they identified that this portion of the serum is responsible for binding with the antigen, and when you are mixing it with the antigen, when this portion you are mixing, there is a precipitation of antigen antibody structure, and then this fraction was reduced when they electrophoresed it. So is it clear now?
Yes, ma'am. It's clear totally. Thank you. Ma'am, I
Okay.
had some other question.
Yes.
Actually, ma'am, I'm still stuck on this antibody. Reason
Okay.
being that I could not face up with the lectures due to some reasons and other
Okay.
examinations. So
Okay.
I wanted to ask that our examination will be on 27th, and
Yeah.
which would be the pattern of exam? Like it will be MCQ based, just like we are giving
MCQ only.
MCQ.
Yeah.
And ma'am, if we need to eliminate certain chapters, so can we take out the last chapters outside because I'm not able to keep up the pace with all these things. I'm having the basics clear, but
Uh, Last chapter means which last week you are telling?
yes, because it will take me a lot of time to come up with these nine chapters. I have skipped a part of that. And then again, this chapter that whole influenza virus. Now, after this session, many problems have been cleared, but still I'm having certain confusion. So I might need to see the videos, some of the videos again. but and then I'm having certain weeks, so like some videos of certain weeks left. So I was thinking about focusing on the basics because I'm having my immunology paper also in my
Okay,
university examination. So
okay.
is it possible to skip certain topics from particular types, or is there any weightage, or everything will be of the same weightage?
Actually, every questions are will be like mix and match kind of. So, from all the topics, more or less one or two questions will be there. So it is very difficult to tell that to skip any topics, but I would suggest you to go through all the lectures only because questions are from actually every topic.
Okay, ma'am. And ma'am, the material that sir has taught and the slides that we have been given, that's enough, no? We don't need
Ah,
to
that
go
is
so much into the.
no, that is enough. If you
Okay.
see sir's slides and sir's lecture, then that is enough, more than enough for the exam. And
Okay, ma'am. And ma'am, experiments like these, for example, I don't think this was much of the importance when I came to the forward part because I thought
yeah,
the main content was, because this was not so important. So we just
yeah.
need to learn the interpretation and not the exact reasons to go in such deep things
No,
because
the exact
time is
reason
not
of
there.
exact reason is just to understand how at that time they did these kind of experiments to like reveal the structure of the antibody. It is not like important to you to in like heart by heart you don't have to learn what they did, who did kind of, but still at least to know how if you are going into a research field after this, then it is better that you should know that these kind of experiments they did so that new ideas can come up in your mind also. So it won't be like you just know the basics like how they have done and all, but if sir has not taught like very deeply, then it is fine.
Okay, ma'am. I just wanted to know about this. Thank you.
uh, any other doubt you are having, you can post in the portal also, or you can ask here also. Two more classes are there, so so if no one is having any other doubt, then thank you for today. So next Tuesday again, if any doubt is there, you can ask.